Let's look at the cell cycle, in which we shall look at the cell cycle itself, the cell cycle checkpoints, and what we call the mitotic catastrophe. So the cell cycle is a self-regulated sequence of events that control cell growth and cell division. And the cell cycle regulators that drive the processes involved in cell cycle include proteins called cyclins, enzymes called CDKs or cyclin-dependent kinases, and an enzyme called anaphase promoting complex cyclone. The primary goal of cell cycle is production of two daughter cells, each with identical chromosomes to those of their parent cell. In rapidly renewing human cells, the entire cell cycle takes about 24 hours, but of course in other types of cells, it can take longer. The phases of cell cycle are basically two. We have the interphase and then the M phase or the mitotic phase. And we shall start by looking at the interphase, which is characterized by a continuous cell growth, and it is divided into three subphases. The first phase, called the gap 1 phase, is the longest and most variable phase of the cell cycle. It starts right after mitosis and continues until the start of DNA synthesis. And in the gap 1 phase, the cell accumulates nutrients. It synthesizes RNA and proteins needed for DNA replication and chromosome formation. In the S phase or the synthesis phase, which lasts for about 7.5 to 10 hours, DNA replication occurs, resulting in the doubling of the cell's DNA and the formation of new chromatids. Then the gap 2 phase involves the final preparation of the cell for division, and it varies in length from cell to cell, from about 1 hour to extended periods. So there are cells that can have an extended gap 2 phase, especially the primary oocytes. During gap 2 phase, the cell inspects its replicated DNA for errors, and the cell continues to grow, and also reorganizes its cytoplasmic organelles in preparation for mitosis. After the interphase, we have the mitosis phase or the M phase. In this phase, the cell divides to produce two identical daughter cells. And this phase lasts for about an hour, and it includes division of the nucleus and division of the cytoplasm, called cytokinesis, and the division of the nucleus is called karyokinesis. The M phase is completed once the two daughter cells are fully separated. Mitosis involves several stages that ensure the proper distribution of chromosomes to the daughter cells. And these phases include prophase, metaphase, anaphase, telophase, and cytokinesis. And this has been covered in the video of cell division. Then the cell cycle checkpoints is regulated by several internal checkpoints that ensure the cell is ready to progress to the next phase of the cell division or cell cycle. And these checkpoints are critical for maintaining the cell integrity and preventing errors. The checkpoints include the gap 1 checkpoint, where the cell is assessed for its size, nutrient availability, and DNA integrity before it enters the synthesis phase. Then the gap 2 checkpoint, where the DNA is assessed for completeness of replication and damage. The M checkpoint ensures proper chromosome alignment and attachment to spindle apparatus before the cell divides. Then the restriction point, also called the point of no return, is the most important checkpoint in the cell cycle. And here the cell self-evaluates its own replicative potential before entering the S phase, or the next phase of cell division, or whether to stop cell division and leave the cell cycle. The gap 0 phase, also called the non-dividing state, is the phase that is outside the cell cycle. And if the cell does not pass the restriction checkpoint, it may enter the G0 phase, where it undergoes terminal differentiation or remain quiescent. The mitotic catastrophe occurs as a result of malfunction of any of the checkpoints discussed earlier. Mitotic catastrophe is defined as the failure to arrest cell cycle before or at mitosis, resulting in abnormal chromosome separation. Under normal conditions, death in these cells will occur by activation of the apoptotic cycle. So when an abnormal chromosome separation occurs, under normal conditions, apoptotic cycle is activated and the abnormal process is terminated. Cells that fail to execute the apoptotic cycle are likely to divide asymmetrically in the next round of the cell cycle. And this leads to the generation of aneuploid cells, that is, cells containing abnormal chromosome numbers. So the mitotic catastrophe may be regarded as one of the mechanisms that contributes to oncogenesis or tumor cell development. This diagram summarizes the cell cycle. So in blue is the interphase, and then in green is the M phase or the mitosis phase, and then of course the cytokinesis. And this table describes what happens in each of the phases of the cell cycle. Thanks for listening and watching. Please like, subscribe, and share.